To include one, returning any excess interstitial fluid to the cardiovascular system, and two, it has a major role in protection of the human body. It does this on a short-term basis via phagocytosis, also known as cell eating, and on a long-term basis via antibody production.
The main purpose of the lymphatic system is to defend our bodies against disease. Most of us stay healthy despite daily contact with various disease-enabling pathogens. We fight these bacteria and viruses through a process called resistance. In contrast, a deficiency in resistance is called susceptibility. There are two types of resistance. One, nonspecific resistance, or innate defenses, which provides basic protection from a broad range of pathogens. Two, specific resistance, or immunity, is learned by the body and constitutes activation of specific lymphocytes to ward off specific invaders. The lymphatic and immune system is responsible for specific resistance. Let's take a closer look. The lymphatic and immune system is made up of fluid called lymph, lymphatic vessels, and other organs and structures containing lymphatic tissue. Lymphatic tissue is a reticular connective tissue containing lymphocytes. Most components of blood plasma are filtered to produce interstitial fluid. Interstitial fluid that enters the lymphatic vessels is called lymph. The only real difference between interstitial fluid and lymph is their location.
The lymphatic system has vessels that run parallel and alongside arteries and veins of the cardiovascular system. These vessels begin as lymph capillaries, which are right next to cardiovascular capillaries. A normal amount of fluid leaves the cardiovascular capillaries and is taken up by a lymphatic capillary, and at this point, the fluid inside a lymphatic capillary is called lymph fluid. Then that lymph fluid continues to travel as the lymphatic capillaries merge to become a lymphatic vessel. These lymphatic vessels usually enter at least one lymph node carrying the lymph fluid into it. The lymph node, sometimes referred to as a swollen gland, which is not really a gland at all, is a lymph node, consists of follicles, which are the structural unit of the lymphatic system. Inside of these follicles are germination centers for lymphocytes and macrophages. Upon leaving the lymph node, lymphatic vessels typically merge to form a lymphatic trunk, which then merge to form collecting ducts, which empty the lymphatic fluid back into the cardiovascular system.
The three primary functions of the lymphatic and immune system are draining excess interstitial fluid, transporting dietary fibers, and protecting against invasion by carrying out immune responses. Lymphatic vessels start out as lymphatic capillaries, located between cells throughout the body. They unite to produce larger lymphatic vessels, carrying lymph to and from the lymph nodes. Lymph flows from lymph capillaries to lymphatic vessels to lymph trunks to the thoracic duct, and then through the subclavian veins into venous blood. The exact same pumps that assist in returning venous blood to the heart aid in the flow of lymph. These are the skeletal muscle pump and the respiratory pump. Contractions of the skeletal muscle compress lymphatic vessels to force lymph toward the subclavian veins. Pressure changes during inhalation also help to maintain lymph flow. From the high pressure abdominal region, lymph flows toward the low pressure thoracic region. During exhalation, valves stop backflow of lymph. There are both primary and secondary lymphatic organs and tissues dispersed throughout the body. The primary lymphatic organs and tissues include the red bone marrow and the thymus. In these areas, stem cells divide to become immunocompetent or capable of immune response. The secondary lymphatic organs and tissues include the lymph nodes, spleen, and lymphatic nodules or follicles. These areas are where the majority of immune response takes place. The thymus is a bilobed organ found in the mediastinum between the aorta and the sternum. Each lobe is enclosed in its own connective tissue capsule, and a layer of connective tissue holds the two lobes closely together. Extensions called trabeculae infiltrate inward and separate the lobes into lobules. Each lobule features an outer cortex and a central medulla. The cortex contains a considerable number of T cells, as well as scattered dendritic cells, epithelial cells, and macrophages. Of all the developing T cells in the cortex, only about 2% survive. The medulla contains more mature T cells, epithelial cells, dendritic cells, and macrophages. Clusters of epithelial cells called thymic corpuscles may serve as sites of T cell death in the medulla, although this is not certain.
This model shows lymphatic vessels coursing alongside cardiovascular arteries and veins, and the lymphatic vessels are carrying the lymph into lymph nodes. A common area to actually feel swollen lymph nodes is under the chin, and is commonly referred to as swollen glands when one has a cold or infection. But actually they're not swollen glands, they're swollen lymph nodes. The reason they're swollen is because large numbers of macrophages and lymphocytes are actively growing.
Groups of egg-shaped lymph nodes are scattered along lymphatic vessels throughout the body. Lymph enters the node through afferent lymphatic vessels. The lymph is directed inward by the afferent vessel's valves, which open toward the center of the node. It then flows through a series of channels called sinuses. These include the subcapsular sinus, trabecular sinuses, and the medullary sinuses. Lymph is then carried away from the medullary sinuses through one or two efferent lymphatic vessels. The lymph, as well as secreted antibodies and activated T cells, are directed outward by the efferent vessel's valves, which open away from the center of the node. Ultimately, the key purposes of the lymph node are to filter out the foreign substances in the lymph and increase the number of plasma and T cells. The spleen, which is found in the left hypochondriac region between the stomach and diaphragm, is the largest single mass of lymphatic tissue in the entire body. It is 12 centimeters or 5 inches in length. The spleen's surface is smooth and conforms to the diaphragm's concave surface. The parenchyma of the spleen features two types of tissue, white pulp and red pulp. White pulp is lymphatic tissue arranged around branches of the central arteries. In the white pulp, B cells and T cells carry out immune functions, while spleen macrophages destroy blood-borne pathogens. Red pulp is made up of blood-filled venous sinuses and splenic cords. The spleen performs three functions within the red pulp. 1. Removing defective blood cells and platelets. 2. Storing platelets. And 3. Producing blood cells during fetal life. Lymphatic nodules are egg-shaped masses scattered throughout the lamina propria of mucous membranes of the gastrointestinal, respiratory, urinary, and reproductive tracts. Lymphatic nodules in these areas are called mucosa-associated lymphatic tissue, or MALT.
The lymphatic system plays a major role in protecting the body. It does this on a short-term basis, as I mentioned, via phagocytosis, and on a long-term basis via antibody production. At this point in the series, if you don't mind, I have to dress for the part, because when I present immunity, it's really a war going on, us against them. This is actually the jacket I wore over 30 years ago when I was a medic in Vietnam. Some definitions first. Immunity. Immunity is our body's defense mechanism against pathogens. A pathogen is any kind of substance that will cause us disease. This can be bacteria, fungi, viruses, or protozoan. Another definition is that of an antigen. An antigen is any living or non-living substance that is recognized as foreign by our immune system. An antibody is an immunoglobulin made by our body that attacks antigens.
Lymphatic tissue starts developing approximately the 5th week of embryonic life. Lymph sacs arise from the developing veins of the mesoderm to produce lymphatic vessels. The paired jugular lymph sacs are the first lymph sacs to appear. They are located at the junction of the internal jugular and subclavian veins. The unpaired retroperitoneal lymph sacs are the next to appear. They appear at the root of the intestine's mesentery. Another lymph sac, called cisterna chyli, develops near the diaphragm on the posterior abdominal wall. The final lymph sacs are the paired posterior lymph sacs, which develop from iliac veins. These sacs produce capillary plexuses and lymphatic vessels of the abdominal wall. Groups of lymph nodes are formed when mesenchymal cells invade the lymph sacs. The spleen is also developed from mesenchymal cells.
All of our body's cells are able to recognize our own cells, i.e. self cells. Anything that is non-self is recognized as foreign and activates the immune response. You can think of our body's defense mechanisms as lines of defenses. The first two lines of defenses are non-specific. It doesn't matter who's attacking us, nobody in particular, we're going to try and destroy it before it does us harm. Our third line of defense is specific, and we'll get to that in a moment. So our first line of defense is non-specific immunity, and that consists of basically surface barriers and mucous membranes. Surface barriers, an example would be intact skin. If our skin is not broken, there's no way that some foreign antigen or pathogen can enter our body. If we breathe or ingest an antigen or pathogen, we depend upon our mucous membranes to destroy it. Both our nasal cavity and oral cavity and pharynx and trachea are lined with mucus to trap foreign particles, and then we can bring them up into our throat in order to swallow it or spit it out. When we do swallow our mucus, the stomach has an acidic environment which destroys the mucus.
Nonspecific resistance to disease is present at birth and aids in the general protection against pathogens and foreign substances. This resistance lacks specific responses for specific invaders, but functions the same way in spite of the type of invader. Your body's first lines of defense against pathogens are the skin and mucous membranes. The epidermis serves as a protective barrier against the entrance of microbes. Shedding of epidermal cells periodically eliminates microbes on the skin's surface. Bacterium seldom breaks through the exterior of the healthy epidermis. Only by open wounds does this usually occur. Mucus, secreted by the epithelial layer of mucous membranes, moistens and lubricates the cavity surface. Mucus-coated hairs in the mucous membrane of the nose catch microbes, dust, and other pollutants. Microscopic hairs called cilia propal inhale dust and microbes toward the throat. This can result in coughing or sneezing. Tears are created and drained away in response to irritants by the lacrimal apparatus of the eyes. The tears are distributed over the surface of the eye by blinking. This helps to continually weaken microbes to prohibit them from settling on the eye's surface. Saliva cleanses microbes from the teeth and mucous membranes of the mouth. Other non-specific resistance mechanisms include 1. The flow of urine to cleanse the urethra, 2. Vaginal secretions to move microbes out of the female body, 3. Defecation and vomiting to expel microbes, and 4. Perspiration to rinse off microbes from the skin surface and break down some bacteria.
Should a pathogen be so fortunate as to get through our first line of defense, we then come to our second line of defense, still part of the non-specific immune response. This includes things such as phagocytosis, cell eating of that bacteria performed by any kind of phagocytotic cell, including neutrophils and macrophages, where we try to eat and destroy anything attacking us. Also, a non-specific defense mechanism includes the inflammatory response. It's normal to have an inflammatory response when we're under attack. There are also natural killer cells that our body have that naturally try to kill anything that is non-self. Also, the production of fever. It's important that our body's thermostat, the hypothalamus, reset and raise our body temperature when we're under attack so that we try to cook and kill any kind of pathogen or antigen. And lastly, we have certain types of antimicrobial proteins, such as complement. Complement is a system of at least 20 non-activated proteins that, when exposed to an antigen or pathogen, become activated and enhance the inflammatory reaction and promote the immune response.
3 main types of antimicrobial proteins prevent microbial growth in the blood and interstitial fluid. These are the interferons, complement, and transferins. Viral infected lymphocytes, macrophages, and fibroblasts produce proteins called interferons, or IFNs. When released, interferons work to stop viral replication. Since viruses can only cause disease by replicating within cells, interferons provide an important protection against infection. The complement system is a group of typically inactive proteins in blood plasma and plasma membranes. Once activated, these proteins complement or augment immune responses. Transferins are iron-building proteins that hinder the growth of bacteria by reducing the quantity of existing iron. Microbes that make it through the skin and mucous membrane are subject to another set of non-specific defense mechanisms: natural killer cells and phagocytes. Natural killer cells, or NK cells, are found in the spleen, lymph nodes, blood, and red bone marrow. Natural killer cells can destroy a wide range of infectious microbes or even selected tumor cells. Phagocytes protect against pathogens by means of phagocytosis. There are 5 phases involved in phagocytosis. 1. Chemotaxis, the moving of phagocytes to the infected area. 2. Adherence, when the phagocyte attaches itself to the microbe or other foreign substance. 3. Ingestion, where projections called pseudopods extend and fuse with other pseudopods to form phagosome, a sac that surrounds the microorganism. 4. The phagosome combines with lysosomes to form a structure called phagolysosome, which breaks down microbial cell walls. The phagocyte also creates lethal oxidants by means of an oxidative burst. 5. The chemical ambush promptly destroys numerous types of microbes. The remaining materials are retained in structures called residual bodies. Inflammation is a defensive response to tissue damage. Pathogens, abrasions, chemical irritations, and extreme temperatures are all capable of producing inflammation. Inflammation features 4 main characteristics: redness, heat, pain, and swelling. A loss of function in the inflamed area can also occur. Inflammation's main functions are to stop the spread of microbes and to prepare the injury for tissue repair.
Now, should a pathogen or antigen be fortunate enough to get through our first line of defense and get through our second line of defense, we only have one line of defense left, our third line of defense, and this is known as the specific immune response. It's either cell-mediated via T cells or antibody-mediated via B cells forming plasma cells to secrete antibodies.
Specific resistance is the body's ability to protect itself against specific agents such as bacteria, toxins, viruses, and foreign tissues. Specific resistance is often referred to as immunity.
The red marrow in our bones are the site of all of our blood cell production. Lymphocytes are produced in the red marrow of a long bone. 75% of these lymphocytes are genetically determined to leave the bone marrow, travel over to the thymus gland, be exposed to thymosin, and be converted into a T lymphocyte, commonly referred to as a T cell. The remaining 25% of the lymphocytes stay in the bone marrow and are termed B lymphocytes, where the B stands for bone marrow.
Two characteristics differentiate immunity from nonspecific response. 1. Specificity for particular foreign molecules or antigens. 2. Memory of previously experienced antigens that aids in a more rapid and forceful response if the antigens should enter once more.
B lymphocytes have specific receptor sites for foreign antigens. When exposed to a foreign antigen that binds to a B lymphocyte, the B lymphocyte undergoes clonal selection and forms plasma cells that secrete antibodies. Antibodies then form specifically against a specific antigen and bind to that specific antigen, trying to neutralize or kill it. T lymphocytes also have specific receptor sites that, when exposed to a specific antigen, T lymphocytes also undergo clonal selection, forming many different types of T cells, the most important of which is the T helper cell. The T helper cell mediates the entire immune response by promoting the inflammatory reaction, the immune reaction, and helping to co-stimulate activated B cells. Without the T helper cell, there would be no immune response. Unfortunately, this is the cell that the AIDS virus, human immunodeficiency virus, destroys.
Immunity has two closely allied responses triggered by antigens. 1. Cell-mediated immune responses, where T cells multiply into cytotoxic T cells that directly attack the invading antigen. And 2. Antibody-mediated immune responses, where B cells change into plasma cells and then secrete proteins called antibodies or immunoglobulin. Antigens are foreign substances that provoke immune responses. The word antigen is derived from the two words antibody and generator. Antigen receptors are found in both B cells and T cells. Through a process called genetic recombination, gene segments are arranged and shuffled to create these receptor molecules. The major histocompatibility complex antigens, or MHC antigens, are located in the plasma membrane of body cells. The key purpose of MHC antigens is to help T cells recognize an antigen as a foreign substance. T cell receptors, or TCRs, identify specific foreign antigen fragments associated with antigen MHC complexes. This acknowledgement is the first signal in T cell activation. Cell-mediated immunity consists of T-cell activation by a specific antigen. Co-stimulation occurs when a T-cell binds to a foreign antigen and receives a second signal at the same time. Cytokines, such as interleukin-2 or pairs of plasma proteins, may act as co-stimulators. Activated T cells grow and begin to proliferate and differentiate into effector cells. In other words, the cells divide several times to form highly specialized clone cells capable of recognizing the same specific antigen. This process occurs in the secondary lymphatic organs and tissues. An antigen binds to B cell receptors, or BCRs, during B cell activation. B cells take in some of the antigen, break them down into peptide fragments, combine them with MHC2 molecules, and then move them to the B cell's plasma membrane. Helper T cells then produce interleukin-2 and other cytokines needed to provide co-stimulation required for B cell proliferation and differentiation. Several activated B cells divide into a clone of antibody-producing plasma cells. Other activated B cells will remain as memory B cells, waiting to react more vigorously should the same antigen return. The majority of antibodies feature 4 polypeptide chains. These are presented in 2 pairs, the heavy chains and the light chains. The tips of the chains are called the variable regions, or the antigen binding site. 5 actions of antibodies include neutralizing the antigen, immobilizing bacteria, agglutinating and precipitating of the antigen, activating the complement, and enhancing phagocytosis. Activation of complement proteins will enhance immune, allergic, and inflammatory responses and assist in clearing antigens from the body. People become more vulnerable to all kinds of illnesses and diseases as they get older. As vaccine response decreases, autoantibody production increases. T cells and B cells become less responsive to antigens, and immune response diminishes.
We've seen that the lymphatic system and its role in immunity is extremely important in preventing diseases. Without our lines of defenses, both nonspecific immune response and immune response, we would certainly perish to bacteria, viruses, parasites, and fungi that normally live on and in our body every day.